then I want to finish with this message. First of all, I mean, probably this is the key message here, that no resistant gene will solve everything. If you want strategies that are long-lasting and are effective, you need to understand the host, the pathogen, and the environment together, and try to make a decision based on that information. The second part, of course, if we want to elevate the resistance and the durability, we need to take in account these elements for a purpose of our crops. Right now, some of the resistant genes that are out there are not long living. So after 3 or 5 years, the resistance is broken. So we need to really increase that durability substantially. I have shown you a little bit some examples of new next generation breeding techniques that we are using that are accessible to everyone, and these are targeting different processes as the sugar acquisition, for instance, and we believe those processes are important to increase the level of resistance of some plants. So we are planning to go with more emphasis in the future about those processes. And one last thing is that coordination is a very important activity, right? I mentioned that pathogens move and normally are regional problems. So if only one country or one breeding program or one national program take decision, that doesn't matter. The important thing is that everyone or the region itself take the similar decision. So that's the only way to contain pathogens in the long term, acting together, basically. And with that, I think I finished. I just want to thank many people that somehow contribute with the slides or information in this presentation and are represented there. And our funding sources are not there. I don't know why, but yeah, it's German government agencies, Gates Foundation, USAID, BBSRC, and we have also money supporting the IRRI CRP, which is a 1CG initiative. And thank you very much for your time.
Thanks, Ricardo. There are a few questions we pasted in the chat box. Do you have you seen in chat?
Wait, I will.
Okay, no problem.
I will stop sharing, right?
Yes, yes.
Chat box.
It's just the green button, beside the green button.
Normally, I have a chat box here. Okay. Okay, I will take one by one. The first one is about neck blast and how that can be introgressed in popular varieties, I would say. I mean, a lot of people are working on neck blast. For all of you, neck blast is caused by the similar pathogen as rice leaf blast. So it's Magnaporte oryzae. How that pathogen infected the panicle is not known, and what resistant genes are tissue specific is not known. But definitely it's coming it's coming a lot in the rice sector now. Regarding the resistance to those, there is a lot of work going on in China, a lot in India as well. We have been screening for neck blast using the 3K panel, the 3000 rice genome. And so far we identified 12 accessions that looks promising. And we are actually now in the crossing part, on the breeding part, to try to develop isogenic lines for that. For instance, we identified one lossy using GWAS in the chromosome 12. We don't know exactly what gene is responsible and how efficient this will be in the field. It's very early to say anything about that, but I think there is some concern and there's some effort about this part of the disease. Can I continue with the rest? Or
yes, yes.
Okay. Yeah, if the SNP or the insertion is enough to block the activation of the sweet gene by the TAL effector. In here is what we know about TAL effectors is that they are not static. They can change and they are a little bit dynamic. So if we introduce one single substitution, maybe the protein is dynamic enough to still bind to the promoter or to the region. This is what we know. So that's why we are selecting small deletions or insertions that are probably 2 or 3 nucleotides in size. So and also the position in the sequence is important. If you block the middle of the recognition sequence, then you have more chance that the binding is blocked. That's what we know. Experimentally, is very difficult to test, but some people are working on that. So yes, I mean the size of the SNP or the mutation is a little bit important. What you don't want is a big mutation or big deletion that change the structure of the promoter itself. That's something that you also don't want. Okay, next question. Neck blast, again, any gene shows similar neck and leaf blast identity? Yes, there's reports for some of the genes that work for neck blast, for leaf blast, and work for neck blast as well. Unfortunately, when those reports are going to the field, they look like the situation is different. So at this stage, we cannot say that particular gene that is good for leaf blast is working on neck blast. Very early to say. What are the limitations of ABR genes? I mean, for a detection process, the limitation on work on ABR genes is that they evolve fast. That's meaning that they change fast. So definitely you need to adapt your methodology to that. Biologically, if that's the question, yeah, I don't know. I mean, the gain and loss, the evolution of these genes is very fast. So as soon as they are selected, they can be in the population, but once they are not selected, they accumulate mutations very fast or can be deleted very fast. That's the only thing. Any novel QTL been identified for rice grain quality in the Philippines varieties? I'm not expert on grain quality. I know that the IRRI, I mean, I know the scientist that is working on that, Nese, and there is a lot of work in progress, in particular in the developing good markers, phenotypic and genomic and markers for selection of grain quality. There's definitely some big progress in that area. But I cannot mention that. I mean, I have no idea. It is possible to do pathogen sequencing at IRRI. Yes, we can do. We are doing nanopore because, for instance, for bacteria XOO, TAL effectors are full of repeats, so we need long reads to try to assemble the genome itself. Right? If you use Illumina or short reads, you cannot assemble the genome, full genome of the bacteria. So definitely you need or either PacBio or nanopore. Now we are using nanopore, It's possible. I don't. That doesn't mean that it's cheaper. Probably it's better to go to a provider that is cheaper than the regular. Okay. Yes, sure.
Yes, you can you answer
continue?
most of them. If you like to continue, you can continue for 2-3 minutes.
Yeah, there's a couple of. How about time? Are we okay in time? Or
yes, for time we are okay.
Okay. So the question is if the ABR genes can work in any other crops. What I can say about that is, ABR is so the secretion of effector proteins by pathogens is a common process now. So we know now that many of the pathogens that have stable interactions with the plants, crops in particular, develop effectors and evolve effectors to adapt to the host. Whether those effectors can move from one pathogen to another is not well known, but definitely several processes are key. So my point is that to suppress the immunity, for instance, of the plant, different pathogens evolve similar mechanisms. So it's a co-evolution process. That's very interesting. So definitely there's cases in which effectors from one pathogen can be used in a different pathogen to facilitate infection. Yes, that's possible. Eloisa Cortez, how do you cope with? Yeah, how do you cope with fast evolving populations like blast? Yeah, that's the whole point of the presentation, is just being as fast as the pathogen. How we do that is very difficult, but one key part is to being able to detect the pathogen early on and to have the tools to do that and the resistant genes in the background. So you need plan A, plan B, plan C, different resistant genes that you can deploy in your varieties in different times. And being able to monitor what is the level of those effector genes in the population. At least for blast, that would be my approach. Okay, one is about the identity of the SNPs. So it should be good. Is a comment like it would be important to make sure that the BLB have an identity test for the SNPs. Yes, definitely. Just to put you in a little bit on the background, what we did is to develop those SNPs is basically go to the 400 genomes that we have and develop SNP markers that are unique for BLB. How we do that? Yeah, it's a very big process in which we have to understand what are the regions in the genome that have recombination process. So you don't want those regions. What are the regions in the genome that are being targeted by selection? So you want those regions. On top of that, you need very conserved genes that are only in bacterial blight, and they're stable across different genomes. So yeah, it's a process of selection until we got those SNPs. Yes, you're right. I mean, cross amplification or cross detection is definitely possible. But we have SNPs in the background that tell us what pathogen may be, if it's a negative or a positive control. Yeah. I think the last question. Oh, yeah.
Yes.
Yeah. How long does the screening takes? Yeah, I mean, for the screening and for blast, yeah, it's a continuous process. Could take a couple of months until you grow the plants and you screen with a representative collection of pathogen. and the right controls, and yeah, a couple of months probably to have a good idea of what is in there.
Yeah. I think it's okay. You have answered. I'll